Good morning, everyone. And we have discussed qPCR system last week. And this week, we will focus on the cell culture systems. So today, we will talk something about basic essentials you need to know in the cell culture system. In this part, we will discuss about the sixth part about the cell culture. First is the principles of the cell culture. And the other part is the stem cells. And the other one is the mycoplasma contamination during the cell culture systems. And then we will discuss something about the transfection, because after the cell culture, usually we will do the transfection to transfect the DNA or drugs into the cells and dealing with the gene expressions or drugs discovery in the whole systems. And then we will discuss something about the cell detections. Cell detections, we will discuss luciferase, cytoactivity, and the apoptosis during the whole systems. And the last one is the troubleshooting. Okay, the first one is cell culture. What is the cell culture? Cell culture refers to a method that stimulates in vivo environment in vitro to make it survival, grow, reproduce, and maintain its main structure and functions. It means we have to stimulate the environment, including the species, appropriate temperature, usually the 37 centigrade, the appropriate pH value, and the certain nutrition conditions. Here, we will show you some cells image. The first one is the primary cells, and the left one is CEF. CEF means the chicken embryos fibroblast cells, and the right one is the mouse liver primary cells. So in these cells, we can see it's generated from the live organism from the animals, but in this case, it will not survive many generations. Maybe 10 times passage will destroy the whole systems. So in this case, the primary cells is just a very short lifetime stage about the whole system. And the other one is the cell lines. Cell lines means it can generate forever, such as HeLa cells. HeLa cells is from a woman who has a cancer about nearly 100 years ago. So HeLa cells is a survivor for already for 100 years, maybe will last forever. And the right one is the 293 cells. 293 cells is from the kidney cells from the human beings. So in this case, the cell lines usually be our materials for cell culture systems or cell biology experiment. Usually we grow cells as adherent cells. In the adherent cells, the growth of the adherent cells divided into the fifth phase. The first one is the free phase. After passaging, so the whole cells is suspended in the supernatant about the whole systems. In this case, the whole cells just collapse into a one cycle. And then the other part is the adhesion phase, usually continuing 4 minutes to 4 hours. And then the cells will be attached to the substrate of the plate, and then it's going into the latent phase. In the latent phase, usually lasts 4 hours to 24 hours. In this part, the cell will do not to divide or passaging. It just do the very active metabolism. It will store the energy to the next level. The first level is logarithmic growth phase. Logarithmic growth phase shows a very active metabolism, and it will divide very quickly. In this phase, it means you can do the experiment at just at this very point of this page. So it means when you do the passaging systems, you have to wait until the next day to do the experiment to make sure your cell culture in the logarithmic growth phase. And the last one is the Platon's phase. In this one, the cells will do not to divide themselves, but to stop. And in this phase, if you make the cells in this phase very long time, the cells will go into death. The reagent for the cell culture, usually the first one is the cell culture medium, such as DMEM or 1640. And the other one is the serum, and the serum will provide a lot of ingredients about the whole systems. And then is the trypsin. Trypsin will do the passaging part about this whole systems. And the next is the penicillin and tropomycin. These two antibiotics will prevent the whole system from the bacterial contaminations. And the PBS as well is the very usually widely used buffer for whole systems. So cell culture medium. Usually culture medium contains a lot of ingredients in the systems. The first one is the amino acid. This amino acid provides a lot of blocks for the proteins, and it is the essential ingredients of all known cell culture mediums. Usually the DMEM and 1640 shows different amino acid formula in the systems. So you have to choose just based on your different kinds of cell lines. And the other one is the vitamins. Many vitamins are essential for growth and proliferation of the cells. And the other one is inorganic salt. The organic salt in the medium helps to retain the osmotic balance and help in the regulating the membrane potentials by providing sodium, such as sodium, potassium, and calcium ions. And carbohydrates. Carbohydrates is the form of sugar are major source of energy. Most of the medium contains the glucose and galactose. However, some contains the mannose and fructose. And the supplement. Usually, the supplement is not survival. It does not exist in every mediums. Some complete growth medium recommended for certain cell lines requires additional components which are not present in the basic medium and the serum. So these components, supplements, have to withstand the proliferation and maintain the normal cells metabolism, like hormones, growth factors, and the signaling substrates. Later in stem cells culture, we will provide a special supplement in the whole systems. And the serum. Serum provides several growth factors and hormones involved in growth promotion and specialized cell functions. It provides several binding proteins, which can carry other molecular into the cells. For example, albumin carries lipids, vitamins, and hormones into the cells. And it also applies to proteins such as fibronectin, which promote attachment of the cells to the substrate. So this one is very important. So during the whole systems, if there's no serums exist, maybe the cells will continue in the, it will stay the suspensions in the supernet. It will not attachment to the substrate. And also the serum provides the proteinase inhibitors, which will protect the cells from the trypsin's digestion. Trypsin. Trypsin is due to his digestivity activity. Trypsin is widely used for cells dissociation, routine cells culture passaging, and primary tissues dissociation. And the trypsin solute is modified with the EDTA and phenol red. The EDTA is used for chelate the cations. The cations such as will help the cells to attachment with the substrate. So with the EDTA will help the trypsin to do the passaging about the cells. And the phenol red shows his pH value. If the pH value is lower than 7, the phenol red will turn red colors. If the pH value is larger than 7, usually the phenol red show is purple. So it will tell us whether or not the pH value is in the right position. And the PBS is a balanced salt solution for is widely used for a lot of cell culture applications, such as washing cells before the association, transporting cells and the tissue samples, diluting cells for counting and preparation of the reagent. Penicillin and tryptomycin. These two antibiotics are widely used to prevent the bacterial contamination for cell cultures due to their combined actions against the gram-positive and gram-negative bacteria. But importantly, these two antibiotics will not influence the fungus and mycoplasma. So these two just can prevent the bacterial contamination. Stem cells. Stem cells is very special cells. It's not like the permanent cells or cell lines. These cells shows very unique characterism about the whole systems. Stem cells are kind of cells that can renew themselves forever, and also it has the ability to differentiation into the variety of the different cell types under the certain conditions. It means it can renew themselves in the certain conditions. It can last forever. And then it can differentiation into many cell types, such as muscles, neurons, or blood cells, or other fibroblast cells. So we can say that human individuals differentiation from the only one stem cells is the one zygote. From the one zygote, the differentiation happens and shows different tissues about the whole human beings. So we can do the classification by differentiation potential about the stem cells. The first of all, we call the totipotent stem cells. The example is the zygote. The zygote shows the totipotent. It means it can differentiation into the whole different kind of tissues about the whole systems. And the other one is the pluripotent stem cells. We call such as blastocyst is the pluripotent stem cells. It can generate all the human beings tissues. And then is the multipotent stem cells, such as neuron stem cells. It only can generate into neural system, not the muscle system, not the bone systems. So it shows little less differentiation potentials than pluripotent stem cells. And the last one is the unipotent stem cells, such as neuron restriction progenitors. So it can just differentiation into the final stem cells, final cells, such as progenitors or neurons. It cannot differentiate into other stem cells. The main applications of the stem cells is to replace, repair, reconstruct, or regenerate the human tissues or organs. Human tissues and organs is a very far aims about the applications. But nowadays we already to create some tissues in the in vitro systems. So maybe not far away we will to replace the human tissues by in vitro systems. Also, the other one is the drug screening. In the drug screening, we can use the stem cells instead of the animal experiment to do the drug screening. So it shows the result more sensitivity and more efficient. Here we provide a lot of production for the stem cell cultures. First of all is the pluripotent stem cell medium, and the other one is we provide the human mesenchymal stem cells medium. So we can provide the two different stem cells mediums. And the other one, this one, Y27632, we call the supplement. We also know that supplement will provide additional functions in the medium. It will provide a very good result for the pluripotent stem cells growth. And the other one, we have cryopreservation medium to store the stem cells or normal cells into the liquid nitrogen. And also we have AP live detection. With this help, we can determine the cells or whether or not have his differentiation potentials. So we will focus on these two very important mediums. First one is the pluripotent stem cells medium. It's completely chemically defined and xeno-free growth mediums. So it means in this medium, there's no serum, there's no any ingredients from the animals. So it's very safe for us to use for diagnosis for drug screenings. So this is the image, the result from the long-term culture effect by our products. Line A is our products, and line B is company A's products. We can see the HPS shows very good result during our products in the cell culture systems. And also the OCT4 gene reference the pluripotency identifications. So in this case, H9 and LT7, there's two different cell lines of the pluripotent stem cells, shows very good result about the OCT4. And in our case, there's 90% about the OCT4 positive cells in the whole systems. And the other one is the human mesenchymal stem cell mediums. So from different derived from the MSC, we can see the very good result continues in our good systems. So the main factors affecting the cell cultures, usually the serum, the culture medium is very important. If you are doing the cell culture very badly, maybe you have to focus on the serum or cell culture medium's quality. It is very important. And the other one is the gas and the pH value, and the temperature and the contamination. For example, the bacteria, fungus, and the mycoplasma contamination. So gas and the pH value. Gas are also one of the essential conditions for cell survivals. And the gas requires mainly oxygen and carbon dioxide. In open culture systems, cells are generally cultured in mixture of the 95% of air and the 5% of the carbon dioxide. Most animal cells require slightly alkaline conditions with pH values of 7.2 and 7.4. Hips combined with the sodium bicarbonate can provide the more efficiency in buff systems, mainly prevent the rapid change in pH. Actually, the hips provide very strong buffer systems, but the carbon dioxide and the sodium bicarbonate provide the strong pH from 7.2 to 7.4. And the temperature. The optimum temperature for human and mammal cells culture is 35 to 37 centigrade. When the temperature is not more than 39, the metabolic intensity of the cells is proportional to the temperature. But the cell will be damaged at the 39 centigrade to 40 for 1 hour, but it can still be recovered. But when the temperature is above to 43 centigrade, many cells will die. So we have to make sure the temperature is very precise. But when the temperature decreases to 20 to 30, maybe the metabolism of the cells will be decreased, but also it can recover. So during the whole systems, we have to make sure the temperature is not too high, but maybe it can a little lower than the 35 or 37 centigrade. And the last part is the mycoplasma contamination. Mycoplasma is a kind of prokaryotic microorganism with the similar bacteria, but no cell walls discovered in 1898. Mycoplasma usually is 50 to 300 in diameter, and they easily can mix into this culture system through the filter membrane. So in this case, the whole systems may be infected by the mitochondria. And this is the image of the mitochondria. We can see this one, this little one is the mitochondria here, mycoplasma. And in the cell cultures, in the incidence of the mycoplasma infection is more than 50%. So it's a worldwide problem to be contaminated by the mycoplasma in cell cultures. In some articles, the numbers is the 68% of the lab is contaminated by the mycoplasma. The mycoplasma contamination is different from the bacterial contamination. If your cells culture systems is affected by the bacterial contamination, the whole system will collapse and the cells will go to die. But in this case, mycoplasma contamination just affects the cell growth, but it does not kill the cells. So in this case, when you observe the cells, you will see the cells can survive during the systems, but it will affect the various of the parameters of the cell membrane, such as influence the cell growth, morphology, genetic levels, metabolism, and the cellular physiology. So in this case, when your system is infected by the mycoplasma, it will affect your experiment result. So in this case, since 2013, Nature journal have formally asked the author must test the mycoplasma contamination if involved cell cultures. So in this case, it shows here, mycoplasma contamination testing status must also be reported. So in this case, we have to focus, pay much attention about the mycoplasma contamination. The detection of the mycoplasma, we have 4 different methods. First one is the DNA staining. DNA staining will stain the mycoplasma's genomic DNA, and it shows very light blue light during the microscope. And the other one is the agar and the bruise culture. But this one is very complex structure, complex systems, and is very vague result when we do the experiment. And the other one is the direct PCR. Direct PCR will detect the genomic DNA from the mycoplasma. So it shows very high efficiency about the detections. And the last one is the luciferase assay. Luciferase assay will recognize the activity enzymes from the mycoplasma. So in this case, it's only detect the live mycoplasma, not the dead mycoplasma. So this one is the DNA staining. So usually with DEPI stems, the circle one is this one, just the genomic DNA of the cell bodies. But in this case, lot of dots, blue light dots, shows there is a lot of mycoplasma contamination in the systems. And this one is the agar and the bruise cultures. We can see the circle one is the different culture showing there is a mycoplasma contamination. And with the direct PCR, we can see specific band about the gene from the mycoplasma. So it shows very efficiency and very sensitivity about the detections. We provide the direct PCR systems and also be very widely used during the experiment. And this one is shows the luciferase assay. Luciferase assay shows also very efficiency about the whole systems and detect only the live mycoplasma contamination. So when we have found whether or not our systems is polluted by mycoplasma, the other one is to elimination of the mycoplasma. We use three different systems. We call the myco 1, 2, and 3, with three special antibiotics to destroy the mycoplasma. And then we can use the very low concentration of these three antibiotics in the supplement about the cell culture. We can prevent the mycoplasma after our system have been elimination of the mycoplasma. Okay. This one is the experimental flow of the cell biology. So cell culture is the basic experiment for the whole systems. After cell culture, the next one always be gene delivery. So we can use the transfection reagent to transfect the gene, the plasmid, the RNA into the cells. And then we can detect the gene expression level to do our experiment. Gene expression level include many levels. For example, this one is the report gene, shows the transcription level. And then the nucleic acid shows the qPCR systems. It can reference the mRNA level. And then we can detect the protein level with the help of the Western blot, ELISA systems. And then the subcellular localization levels, we can use the microscope to detect the proteins subcellular localizations. And then is the protein interaction. And the final part is the cell levels for the whole systems. So we can focus on the CCK MTT to do the cytoactivity or apoptosis is just the cell levels. The last week we discussed the qPCR system is detect the nucleic acid level for mRNA levels, and then the Western level for proteins. And this one is next we will discussing the luciferase system, luciferase assay to do the reporter gene to reference the transcription level about the whole gene expressions. Okay, so after cell culture, we have to focus on the gene delivery. Transfection. Transfection is the introduction of the DNA or RNA into eukaryotic cells to study and modulate the gene regulation, allowing the understanding of the gene functions and the protein expressions within the whole systems. So in this case, there's several methods for transfections. First one is the chemical method. The liposome mediate or non-liposome mediate and the PEI or cation phosphase is also called the chemical method. And the other one is the physical method. We can use the machines to create a very high voltage to do the electroporation. In this case, it's a physical method to put the DNA or RNA into the cells. And the last one, some cells are very difficult to transfect. So in this case, we have our own biological way. We call the viral medium transfections. We can use the lentivirus, endovirus, or retrovirus to do the viral medium transfection. The first one is the liposome medium transfections. The liposome compound with the DNA shows the lipoplex, and the lipoplex will recognize the signals from the cell membranes and during the endocytosis into the cells. This lipoplex is covered by the endosomes. But in this case, the endosome maturation into the lysosome, it will destroy the DNA. So in this case, the transfection is failure. But the lipoplex is escaped from the endoplex. It means the transfection system is worked, and the DNA go into the nucleus and shows the transcription and the protein expression. So in this case, the liposome medium transfection system is our products here, PL transfection reagent. This reagent is suitable for transferring DNA into eukaryotic cells. So unfortunately, maybe it's not very suitable for RNA into the cells. And in this case, it's very good reagent for lung or liver or cancer cells tested in the whole systems. So if you focus on the cancer cells, you can try PL transfection reagent. In this one, this Q7 is a lung cancer cells, and this one is stomach cancer, and this one is the pancreas cancer cells. Okay, so we can see the transfection efficiency from the both cancer cells is very high levels during the whole systems with the help of the PL transfection. The other one is the non-liposome mediated transfections. Non-liposome is not means there's no lipid at all. It shows the very single layer about the liposome. So we call the non-liposome transfection reagent. And this one is suitable for transferring the DNA and RNA into the eukaryotic cells. So you have to transfect RNA such as siRNA, microRNA into the cells. You can use the EL transfection systems. And it shows very high efficiency for different kinds of type of cells. Some is regular cells or permanent cells or cancer cells. It also shows very good results here. And also it's suitable for adherent cells, and the suspension cells also can work very well. And this one is stomach cancer cells. So it shows a very good result here. Or maybe this red signal is not very obvious. From our machines, we can see very obvious the red signals from here. And it shows it's a normal transfection, no liquid change during the whole systems, with the serum or with the antibiotics during the transfection, also shows very good result compared with the normal one. And this one is transfection with the siRNA into the systems. Here it shows the siRNA's result. It shows a very good effect about the gene expressions compared with the normal one. So it shows you can transfect the siRNA into the systems. And this box shows the successfully transfected cells in our lab. And this box is widely changed because a lot of customers shows their data to us about the whole systems. And this one is used electrophoration systems. These machines shows very high voltage, and then it shows very high intensity electrical pulse for several microseconds or several millimoseconds. It will cause destabilization about the system and punch different holes in the cell membrane, and then it can transfect the gene or drugs into the cells. But in this case, the voltage is very important, because if the voltage is too high, it will kill the cells immediately. So we have to optimize the voltage and the times between the whole systems. And the last one is the viral-based transfection. For some very difficult cells, such as primary cells, stem cells, undifferentiated cells, the efficiency of the target gene to transduction can be generally improved. We can choose the viral-based transfection in the whole systems. In this way, we have three different virus can do the job. The first one is the endovirus, and then is the lentivirus, and the last one is the retrovirus. The viral type is different. The endovirus type is the double-stranded DNA virus, and the lentivirus and the retrovirus is the RNA virus. So in this case, there's a genome integration about the whole systems. The endovirus will not convert their plasmid into the genomic DNA. So it's very safe for us to do the human therapy the systems. But the lentivirus shows very highly risk to compile the genes into the genomic DNAs. So during the whole systems, maybe the lentivirus shows a highly risk for human recipe. And also shows very good result about the protein expression level, and the protein expression speed shows very fast in endovirus and is slower in the lentivirus. But in this case, the typical titer is much higher in the lentivirus systems, and the insert size of the capacity is different. The lentivirus and the retrovirus just 8 kb, but the endovirus shows the 30 kb for the target genes. But here, the immunogenicity about the endovirus is very high. So in this case, when you do the animal experiment, we have to focus the immunogenicity, because the whole systems maybe create very high efficiency about the whole systems. Maybe the endovirus shows a little risk about the animal tissues, animal experiment. When we use the lentivirus package, usually we have to do the cloning systems to create the expression plasmid with the gene of the interest of the whole systems. And then with the other 3 package plasmids, the 4 plasmids will be co-transfected into the 293 T cells. In these cells, the lentivirus will package in the cells, and it will separate from the supernatant of the cells culture. In 48 hours to 96 hours, the supernatant will contain a lot of lentivirus. And we can use this one, the lentivirus precipitation solutions, to concentrate the lentivirus, and then we can use the lentivirus qPCR titration kit to determine the titer about the lentivirus. And here, this box shows the comparison of the transfection method between the liposome mediate, the kaolin mediate method, and also the electroporation method and the viral-based method. So we can see the viral-based method shows very high efficiency for the systems, but the work is very difficult for cell lines, and also the procedures is very complex during the systems. But this one, the liposome mediate, is very easy to use, and shows very high efficiency for many cell types specifically. After the transfection, we have to detect the cell detection of the systems, usually the gene expression level. The luciferase assay shows the transcription level about the genes, and the cytoactivity, we can use the CCK and MTT to detect the cell's activity about the systems. And apoptosis shows the cell death about the systems. So first of all, we have to focus on the luciferase report assay systems. The transcription factor is a kind of protein with a special structure and regulating the gene expressions, known as trans-acting factor. And then some transcription factor only by the specific sequence in their target promoters, which are called cross-acting element. So trans-acting factor and the cross-acting element is a very key to the whole systems. And the DNA binding domains of the transcription factors and the cis-acting elements achieve the covalent bindings. And then you can show the ability about the inhibit or enhance the expression of the genes. So we can use a lot of reporter gene system to detect the transcription level about the trans-acting and the cis-acting elements. Here is the common reporter genes, such as LAC-Z, CAT, and the GFP is also the reporter genes can be used to detect the whole systems. But in this case, we can use the luciferase assays. The luciferase assays shows very rapid, sensitive, and widely range, and shows very low intracellular interesting activities. So it means there's very low background signals during the systems. So in this case, luciferase assay shows very sensitivity about the whole system in the reporter gene systems. This is the principle of the luciferase reporter gene systems. Different substrate with the recombinant firefly luciferase and also the renina luciferase. And then the whole reaction will create different light wave of the different colors of light. And it will be detected by the machines we call the luminometer machine, can detect different kind of light to show the signals between the first firefly luciferase and the renina luciferase. So the procedure about the whole system is very easy to use. First one is to move the medium, wash the whole systems with the PBS, and add the cell lysis buffer into the systems, transfer the cell lysis to EP2, and then spin the collector supernatant. And then with the help of the luciferase reaction reagent with the cell lysis, and then the first measurement will detect the first light signals. And then with the luciferase reaction reagent 2, it can detect the second measurement caused by renina luciferase systems. The amplification about the luciferase assay is very widely ranged. To start the receptor activity, transcription factors, promoters, and enhancers can be used. All study of the intercellular signals, such as P53 signals and N of kappa B pathways, we can use the luciferase assay. And also, we can study on the degradation of the RNA by proteins or target of the microRNA. And also, we can study on the protein and protein interaction. And also, we can use the drug screening about the whole systems. Cytoactivity, we can use the MTT and the CCK to detect the cytoactive. MTT system is a very classic ways to detect the quantitatively measured of the cell death during the cell culture are crucial in any experiment involving the cell lines or whole cellular clinical samples. In this case, such as CHO cell lines or viral cell lines, we have to use the MTT to detect the cytotoxicity effect and the compound with the experiment to detect their variability activities during the whole systems. But the CCK can detect more sensitivity than MTT. When you use the MTT to detect the systems, MTT only involves the mitochondria, the hydrogenase. And then in CCK, CCK8 is far more sensitivity than MTT assay. And then the WST8 formation is created by CCK, is water solute. It's not that from the crystal like the MTT. If you use the MTT, the crystal formation is have to be dissolved by DMSO, but DMSO is toxic for human beings. So in this case, we can use the CCK is more safer for ours to operate the systems. And then you can use the machine to measurement the OD at the 450 nanometer to give the number of the variable cells. And the application about the CCK is to do the cell proliferation and the activity assay. For example, it can to detect the growth factors and the cyclins. And also the other one is the cytotoxicity assay. It can do the tumor necrosis factor alpha effects, such as whole systems, and also can be used drug actions, drug screeners for biological components in the systems. And our reagents shows very low cytotoxicity about the whole systems. Compared with other companies' reagents, after the 24 hours incubation, the image shows there's no significant change during the whole system's incubation system. And it's very easy to operate. First of all, to add the CCK into the supernet at the ratio of the 10% of the supernet, and then incubate for 1 to 4 hours, and then use the machine to detect the signals. So it's very easy for people to use. The last part is the apoptosis. Apoptosis is known as a programmed cell death, PCD, which is the regular and controlled mechanism of the growth and the development of the organism. So it's also called the cellular suicide. In this process, the cells itself take part in his death. And then we have to tell the difference between the apoptosis and the necrosis. The necrosis means the whole system is be destroyed by outer force, such as toxic or others affected by the external agents like toxic trauma and the infections. Apoptosis is a highly regulated, so in this case shows totally different steps about the apoptosis and the necrosis. This one is the necrosis, so we can see the cellular is collapsed, and also the whole content inside the collapse will be destroyed and released into the supernet. But this one is the apoptosis. In the later stage of the apoptosis, there is not the membrane collapse. It just shows the apoptotic bodies in the whole systems. So it's a programmed cell death. The processing of the apoptosis can be divided into different steps. The first one is the cytoplasma shrinking and the junction among the cells disappear, and opening the mitochondria permeability of the transition pore, membrane potentials, and the cytochrome C release into the whole systems. And then there's membrane labeling, and also the PS translocation from the inner leaflet to the outer leaflet, but without losing the integrity about the cells. So in this part, this one, we can use the annexing 5 system to detect the early stage of the apoptosis. In the later stage of the apoptosis, the nucleus collapse and the chromatin and the DNA cleavage into the oligonucleosome length DNA usually is 180 to 200 BP. And in this case, we can use the such as tunnel system or DNA dining to detect the later apoptosis about the whole systems. So here we can provide the different method to detect the apoptosis. First one is the cellular morphology. So you can use the microscope to detect the cellular morphology. And the other one is the DNA staining. Also, we can detect the nucleus situation to detect whether or not the apoptosis. And we use the mitochondria membrane potentials, JC1 assay. We can detect the mitochondria's integrity. And then the BCL2 collapse, these signals is the apoptosis related proteins. We can use the ELISA and the Western blot to detect the signals here. And then we can use the annexing 5 system to detect the state of the cell membranes. And also the last one is the integrity of the nucleic acid. We can use the tunnel systems to detect the later stage of the apoptosis. Okay, the last one is the cell culture troubleshooting. These 3 questions is the most frequently we will ask during the cell culture systems. First one, how to replace the cell culture medium or serums, which is different from the original one. So we call you can choose the 25% step by step. Maybe the 4 times later, you can replace the new cell culture medium into the systems. The other one is that it is necessary for antibiotics to be added into the culture medium. No, if you have a very good experiment behavior and you can control the environment, you cannot add the antibiotics into the systems. And the last one is what is the centrifuge speed for the collecting animal cells? Usually is the 800 to 1000 RPM to collecting animal cells. How to store FBS? FBS always restore at the -80. So when we use the FBS, we have to throw the FBS. First of all, we have to remove the serum from the freezer and allow it to throw at the refrigerator at 2 to 8 centigrade overnight. You can regularly mix during whole procedures. Maybe you can upgrade the systems by leave it in the room temperature. But warning here, do not incubate FBS at 37 centigrade. Usually, clients throw the FBS in the 37 centigrade. It will destroy the components in the serum and destroy the whole systems. And avoid repeating, freezing, and throwing the serums. After throwing, it can be adequate into small portions and placed at 4 centigrade in the refrigerator. Okay, the last few slides, we will discussing some phenomena here. First one is cell grows very slowly. The possible cause about that is maybe the growth medium temperature is not correct. So you can use a pre-warmed growth medium, such as recommended by the suppliers. Usually it's the 35 centigrade or 37 centigrade. And the cell's seedling density is too low. You can increase the seedling density. Maybe 1 plate to 3 plate is enough. And then the other one is the cell culture is contaminated by mycoplasma. So you have to discharge cells medium and the reagents, or you can use our reagent to do the elimination about the mycoplasma contamination. And the other one is cells have been passaged too many times. We have to recommend that you can use healthy, low passenger numbers cells. And the last one, maybe the serum or the medium shows very poor qualities. So you have to choose the serums in very good quality controls. Cell death. Maybe the carbon dioxide density shows incorrect. Usually you have to increase or decrease the percentage of the carbon dioxide in the incubator at 5%. Maybe you can change it a little bit. And then you can add the sodium bicarbonate concentration should be 20 to 33 to 7 gram per liter. And maybe you use the trypsin over digested cells. You can change the digester in very short time, maybe 1 or 2 minutes, that's enough. And do not use the less trypsins as well. Or if your cell lines is very sensitive, you can use not use the trypsin. You can use the EDTA instead the digestive steps. The last part is the bacterial, yeast, or fungus contamination. So we have to describe the culture and medium. The bacteria or the fungus contamination is very difficult to elimination. So in this case, maybe you have to choose a new culture systems. And maybe there's inefficiency buffering systems. So you can see the colors of the supernatant of the systems. If it turns yellow or purple, is instead of red, it means the pH is different. So we can use Gibbs buffer to the final concentration of the 10 to 25 micromole to restore the pH value. And the other part is the low transfection efficiency. In this case, maybe the many possible cause is plasmid DNA, siRNA, and transfection reagent dilution ratio in this media is different. So maybe you can optimize the DNA to transfection reagent ratio to 1:2 and 1:3. And then maybe we have to focus on the dilution times or complex incubation time. Maybe this time is too short. Always clients will just mix the incubation just for 5 minutes, but the 5 minutes is not enough. We have to add the dilution reagent together into the incubation systems for 20 minutes at least. And maybe there's not enough plasmid DNA or the whole system shows very poor qualities about the DNA or RNAs. So you can use the nanodrop to detect the quality about the plasmid DNA or RNAs. And then maybe the cell's condition is not so good, or the cell aging or endotoxicity is also shows in the systems. So we have to adjust the status of the cells. We have to make sure there's no endotoxin plasmid in the transfection systems. And then the next one is the cell density was not optimal. So we have to make sure the cell density in the transfection systems is 70 to 90% about the system. And the last one, maybe there's storage failures. We have to make sure the whole systems about the transfection reagent do not freeze the transfection reagent. You only store the transfection reagent at the 4 centigrade. So focus on the centigrade about the restorage conditions. Okay, this is today's speech. I hope this information will help you in your latest experiment in cell biology experiment. Thank you. So if there's any questions? Okay, here we see 3 different questions. First, what will happen if you use a different cell culture medium than recommended? Will it influence the gene expression or only the growth rate? It will influence the gene expression. Normally, we will normally, we have to focus which different kind of cell medium is for which type of cells. For example, DMEM is different from the 1640 medium and shows different. So when you use different cell lines, you have to focus on the different cell mediums, especially for stem cells. These are very specific cell mediums. And also it will influence the gene expressions, also affect your result of the experiment. The next one, why does some cell culture medium have L-glutamine and some don't? Are there disadvantages having L-glutamine in your medium? L-glutamine is a very important ingredient in the systems. Usually the fresh mediate and the serums do not use L-glutamine added extrally. So in this case, you can just use the fresh media and the serums directly. But if you store the medium at the 4 centigrade for a very long time, for just like 4 months, in this case, you have to add the extra L-glutamine into the systems. Nowadays, we can say there's not very specific disadvantage when you use the L-glutamine. So it's very safe for us to use. how to clean the original tissue from the host. Always you can use different buffer to wash the tissues, such as PBS or Hanks buffer can be used for the original tissue from the host. You can wash maybe 10 times of the liquid. There will be bacteria or others on the original tissues. How do we remove the bacteria so that we have the resistance culture will be not infected? We have to use the Hanks buffer or PBS buffer with a very high concentration of the antibiotics to wash the tissues. In this case, we can remove these bacteria. So maybe the concentration is 10 times than the normal one in the cell cultures. In this paper, it is reported that we have to use the trypsin as dissociation reagent can alter the phosphorylation status of the certain molecules. Do you recommend using trypsin or dissociate cells for the cell signaling studies? Yes, because the trypsin is enzymes. It shows very high strong activity about the cutting the proteins which exist in the cell membrane. So if you study the cell signaling study, it means you have to study the proteins exist in the membrane. In this case, do not use the trypsin. You can use the EDTA instead of the trypsin. But maybe when we use the EDTA, it have to incubate for a very long time. For trypsin, maybe you can incubate for 1 to 2 minutes. But when we use the EDTA, maybe you have to incubate the system maybe 10 to 15 minutes. But it will not influence the cell signaling studies. Okay, the next question. Which method of the transfection give higher transfection efficiency? We recommend the liposome and the non-liposome method for transfection systems, such as PL and EL transfections. But for stem cells, for primary cells or non-differentiation cells, we can use the viral medium systems, such as lentivirus system, to do the higher efficiency for transfection. The next question, it is possible to look at the contamination by looking at the medium or the flux. If we encounter the bacterial infection after observation under the microscope, but the pH was basic and no tubuling from the medium can be seen, the cells and alive too. If you are affected by the bacteria or the fungus, you can see from the microscope directly. But if your system is polluted by the mycoplasma, it's very difficult to look at the medium of the flask from the microscope. So if you're affected by the mycoplasma, you have to use the PCR or luciferin system to detect the mycoplasma contamination. But if the bacteria or fungus contamination, you can use the microscope to see the whole systems. May I know which aspect I can look at the source of the determined type of contamination? Oh yes, it shows different phenomena about the fungus and the bacteria and the mycoplasma. So mycoplasma, you can use the different reagent to detect the mycoplasma contaminations. When it shows the bacterial contamination, the whole systems will be not shows very transparent. It shows like a lot of seeds in the whole systems. But when the fungus contaminations, you will see the very large fungus. You will see the fungus just like a flower floating in the whole systems. It's very not good looking in the whole systems. The next question, what's most of the psychophysical relation passing member? Usually the primary cells usually have to maintain the passenger number lower than 10 passenger number. And the cell lines means you can just control the cell passenger number at the 50 passenger number will be okay. If we placed extra drug MTT, what can we use as a solvent? DMSO can be used as solvent to dissolve the MTT. To detect early apoptosis, if we don't have the flow segmenter, what do you advise to use? You can use the chromosome C assay to detect early apoptosis instead. Current, we are using the CLAPS activity as an indicator of apoptosis. Yes, the CLAPS 8 activity also can represent the early stage of the apoptosis, but the chromosome C also can detect the resistance. Also, JC1 assay can also be used for detect the mitochondrial integrity. So this assay also shows the also reference the early stage of the apoptosis. What's the maximum and the optimum passenger number for cells? 50. 50 is the maximum number about the passenger number. So if you are close to 50 times, it means you have to get rid of the cell lines. In cell culture, always got problems with the presence of the black swimming dot. What's your suggestion to prevent it from happening? And what's the specific antibodies we use? Black swimming dot. Black swimming dot is here. I know that part. 黑胶虫怎么说？黑胶虫。Black swimming dot is it's very difficult to get rid of the black swimming dot, and there's no specific antibiotics can be used. So in this case, you就是黑胶虫，黑就是黑色的黑，胶是胶体的胶，虫是虫子的虫。对。对呀，这是胶。对。So in this case, maybe you have to get rid of the cell lines. How to prevent this situation is you have to. In this case, you have to control the environment about the whole systems and make sure you have the very good behaviors about the operation systems. So maybe the black swimming dot is very difficult to get rid of. In this paper, it is reported that we use the trypsin as. Oh, okay, I already answered those questions. How is the sensitivity of the CCK8 compared to those fluorescence and luminescence based? CCK8 is much more sensitivity than MTT systems. And also the GFP and the LAC-Z shows very low sensitivity about the system. So nowadays CCK is widely used in the systems for detect mental activity. What's the physiological relevant? Already answered that. Detect early. For FPS from -20, you should not throw in the water bath. Instead, correctly to. Yes, when you throw the FBS, do not put at the 737 at all in the whole times. in the 37 centigrade, it will destroy the ingredients and the various essential component in the FBS systems. So do not throw at 37 centigrade at all. Just put it at 40 centigrade. It will be okay. Okay, I think I answered all the questions. There's a new one here. Following up with the trypsin, have you used activities or ingredients to replace the trypsin use? EDTA can be used to replace the trypsin use. EDTA, yeah, EDTA butter. In maybe I think is the 20 micromole concentration for EDTA butter can replace the trypsin. Okay, I think I already answered all the questions.
Thank you, Dr. Zhao, for the Q&A session. So, is there any other questions that you would like to ask Dr. Zhao regarding the cell culture?
Excuse me, I just would like to find out the CCK detection method as compared to MTT assay. Do we need extra or different equipments to detect it, right? Compared to MTT assay based on the ELISA plate reader.
Beg your pardon? I'm not quite follow that.
Okay, for CCK detection method for cytotoxicity activity. Okay, just now you show the slide, right, that we had to incubate for 1 to 4 hours. After that, we have to read, read for the plate. That that detection can be read by the normal ELISA plate reader, right? Or there's another.
The normal one will be okay.
Normal ELISA plate reader that MTT assay will do.
Yes.
Okay, great.
Just to detect the OD at 450 nanometers.
Yeah, it's about like MTT assay as well, right?
Yes, yes.
So how?
Another one, 1 to 4 incubation time maybe can be reduced. So maybe if the color shows very deep in the systems, maybe you can reduce the time to 30 minutes will be okay. Yeah.
Okay, minimum 30 minutes, is that?
Yeah.
All right. So how about the cost of reagent compared to MTT assay?
The cost of the reagent, I think is similar to MTT, I think.
It's about the same.
Yeah.
All right. Thanks for the information. Yeah, because it's more specific, like you said, right, compared to MTT.
Yes. And it's much safer for people to operate the system. Yeah. Because DMSO is very toxic for human beings. Yeah.
Yeah. Because of that, I think I'm thinking of changing that for my students to use that. Yeah, thank you for that. Yeah.
Okay. You're welcome.
So, is there any other questions on the floor that would like to ask Dr. Zhao?
Dr. Zhao, the black dot that you mentioned, 黑胶虫, is it called nanobacteria?
No, actually, in this case, we not quite sure about what the hydrogen is. Yeah, it's this one. But some people think it's organic tissues, just a very small tissue. It's not the living organism. But some people think the hydrogen is just something like bacteria. So it's not get very convincing data for these ingredients here. But it's very difficult to get rid of. So if you have been infected by these black swimming dots, you have to get rid of the cells and clean the environment and show very good behavior of the procedures and use the new cell lines to do the next experiment.
So
Can I have another general basic questions? Because I'm beginner for the animal cell culture work. Okay, for the thumb rules, how about the passage of the animal cell culture that I mean suitable for any experiment? Is there any basic numbers of the passage that we should apply or should practice for any animal cell culture work or experiment?
是那种传代的次数吗？I don't quite follow your questions. Is that you asked how many pathogen numbers for the system?
Yeah. How
no
many
more than
times
10.
you can keep subculturing of the animal cell for any experiments? Yes.
Yeah, 10 passages will be great.
Not more than 10, is it?
Usually not more than 10. Yeah.
If more than 10, then maybe
maybe a little effect, but maybe the result can also be get. But we recommend not more than 10.
I see. Okay, so that's the thumb rules, not more than 10 for animal cell culture passage, right?
Yes,
if you want to use
yes.
for the good
but in
response.
some circumstances, maybe the maximum of the pathogen time is 50. The
50.
maximum is 50. Yeah, but
Okay,
we recommend 10.
Okay, may I know what types of cell culture that can go up to 50 and what type of cell culture that can, is better to
It means if you have a lot of storage for your cell culture, 10 will be okay. But the cells is very precious, so you have no more cell lines for the system. So maybe 50 is the maximum time.
Yeah. But as long as the cell culture are not mutated, or still can see very good intact morphology of cells, shouldn't be a problem, right? If up to 50 time of passages.
Yes. You can choose. Let me see this one. We have to make sure the cells by his circumstances, not just by the number of the passages. If the cell lines shows very good quality about the system, you can use it, not just based on the numbers of the passenger.
So in this case, how can we determine the cell still, appropriate for the experiment if let's say more than 10 times of the passages? What kind of observation that we have to check the cell?
You can check the phenomena about the whole pathogen situations, such as this part. You can detect the different phase about the whole system. So you can see there's anything strange things during the whole systems for adhesion phase or latent phase or logarithmic growth phase during the whole system. So you can detect whether or not this cell lines is suitable for experiment later.
All right. Thank you very much, Dr. Zhao, because I joined in quite late just now. So sorry, yeah. I think
It's
you already
okay,
explained
it's okay.
earlier.
Yeah.
Okay, thank you very much, for your sharing.
Okay,
Yeah.
you're welcome. There's another question. Can we use your stem cell medium to maintain the cancer stem cells? Yes, you can use that. You can use the chlorine-prudent stem cell medium to maintain the cancer stem cells. But you have to try first. We were not sure you can maintain all types of cancer stem cells. The normal one can be working. Okay. 50, not 15. 50 for pathogen number. 50. It was stated just now. Since Q2 not did stress that after incubation. We use this cancer entity declaration after the incubation. Yes, when you use CCK instead of MTT reagent, the plates can be read right away after the incubation period. So after the incubation, you can read the plate in the machine directly. Okay.
Dr. Zhao, you mentioned the cytochrome C assay and JC1 assay. So are these ELISA based? Because the JC1 assay that we used before, we have to use a flow cytometer.
No, don't quite follow. These two assay refers
Okay,
what?
for JC1 assay and cytochrome C assay that you recommended for early apoptosis detection,
Yes.
are
yes.
these two assays used like a ELISA reader?
No, not use ELISA reader. It's use specific machines to detect JC1 assay. JC1 assay is to detect the voltage between the inner membrane and outer membrane of the mitochondria. So you need to use a different kind of machines. And the mitochondria C, you can use the ELISA or Western blot to detect the mitochondria C. Yeah.